pigment. Pigment. That's what we're doing. We're going to do a demo lab on pigment. So what is pigment? There's a picture back here of a thing called chromatography paper. Chromatography paper is paper. It's made of cotton, so it's absorbent. Takes up water. Water works really well for a lot of things. We have water, and we also use this, which is alcohol. The reason that we use alcohol is because pigment that we find in plants is a little bit fatty. He's fat in plants. Plants are fat. No, you're fat, Fritsche. Plants are not fat. But pigment has a little bit of a lipidy substance to it, and so with alcohol, they don't blend very well, and so it pushes the chlorophyll away from it. And some pigments actually are more afraid of alcohol than others, and so sometimes you can see them better than others. This picture right here behind me, this was definitely done with an alcohol, and you can see the different the different pigments that are traveling here, and I'm going to do a little bit of a demo just to kind of show you what's going on with these pigments and how they work. What does chromatography paper do? Here's a little hint. Chromatography helps to separate pigments, pulls them away from each other. And we're going to take a look at a couple of different pigments. Now this is an example of chromatography paper. I cut this out. Again, it's like a cotton-based thing. I folded it so that it will stand up straight in the beaker. And what we can do with this is we can put different things that have pigments in it. This is a Sharpie. A Sharpie is water soluble, and so what I did was I took this Sharpie and I drew a little line at the bottom of this. You'll notice that it's a black Sharpie. Black, if you'll remember about pigments, black is all of the colors of light being absorbed, so you don't see anything. That's why it is black. It looks dark because, well, no light is bouncing off of it. So I took a black Sharpie and I put the black Sharpie on this chromatography paper, and you can see here, where's my camera? There's my camera, okay. You can see here on this, it's not focusing very well, is it? That I have separation of a couple of different colors. It mostly looks like a purplish color, but you can tell that there are different colors in that Sharpie. If it was a different marker, I think a more water-soluble marker would have definitely seen the colors separate a little bit better, but this is the idea. We're separating pigments. That's the whole idea behind this. This here is a battered and bruised and hurting, what is this? It's a leaf. Spinach leaf, okay? Pretty hurting. But what I did was I took another piece of chromatography paper and I rolled a penny over top of this piece of spinach onto the chromatography to transfer paper to transfer the pigment over to the paper. And what we got here is this. This is, again, the new chromatography paper. Now what you can see is you can see two different colors really. Technically, there's three different colors in there. There's an orange, there's a yellow, there's a green. You can't really see the bluish green, or yeah, you really can't see the bluish green very well. You see more of the olive green that's in there. The different colors, and you can see from the board right here, we have chlorophyll B. Chlorophyll A is very difficult to see. It's really hard to see the bluish green, and the reason it's really hard to see the bluish green is because that's one of those pigments that the fat doesn't really stick, or not stick, the opposite, push away from the alcohol as well as the chlorophyll B does. But you can see the chlorophyll B, nice green color. You got a bluish kind of color here. This is xanthophyll, and this is carotene or carotenoids. Xanthophyll usually travels a little bit further. It usually travels past the carotene, but you get the idea that this is a yellow color and that's an orange color. So we have a couple more things that we're going to add to this video. Chlorophyll B. Who? Oh, Shoshaki. Sasha, what's going on? Okay, so why is chlorophyll B green? Well, this color green, the reason it's green is because green is hitting it, right? Green is bouncing off of it, so we can physically see it green. All the other colors are being absorbed by it. Chlorophyll A, this is taking a combination of the blue and the green, and blue and green are both bouncing off this, absorbing everything else. With xanthophyll, xanthophyll is that yellowish kind of color. Yellow is is reflecting, and every other color is being absorbed by that. Same thing with carotene or carotenoid. I like carotenoid better. It's the orange and the red. The orange and the red, those are being reflected, whereas all the other colors are being absorbed. The pigment's job, hint, hint, wink, 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 the pigment's job is to absorb light. We just saw that when we did photosynthesis, and we did the whole entire thing on the light and how the electron is being excited and all that fun stuff. And so the purpose of the pigment is to absorb those electrons so that those electrons can be excited. So that's the purpose of the pigment. In this picture right here, we can say that carotene or carotenoid, this is the one that's traveling the furthest. This is making its way all the way towards the top. Sometimes I say xanthiville. It's hard to tell the difference. But in this picture, we say carotene, so we're going to say carotene. Okay. Chlorophyll A is reflecting a certain color. Here's the chlorophyll A. This is reflecting blue. This one's reflecting green. This is reflecting yellow. And this is reflecting orange. Okay. So that's the demo lab. You saw chromatography paper. Chromatography paper separates pigments. Okay, the reason that we use those different pigments, they're absorbing different colors, and I think that's about it. So there's your video, there's your demo lab. Now go take the quiz. I'll see you on Tuesday. That was for you, Trevor.